Welcome to today's masterclass. I am so excited to guide you through this explainer because today we're diving into one of the absolute most phenomenal defense systems in existence. We are going to look at how our bodies literally manufacture microscopic guided missiles just to keep us alive. These missiles, we call them antibodies, and the high-tech factories that produce them are our B cells. It's a story packed with precision, danger, and just incredible biological engineering.
So, here is our syllabus for today. We'll be covering the B-cell academy, passing the tolerance test, activation and T-cell help, the anatomy of an antibody, and finally, we'll wrap up with effector functions in action. Let's get right into it.
Starting off with Section 1, the B-cell academy. Because, to get a highly specialized immune defender, you absolutely need a rigorous military training ground. And for our main character, the B-cell, that training ground is the bone marrow.
But if we look chronologically, the whole process doesn't actually start there. Early on, B cells originate in the yolk sac, and then they transition over to the fetal liver, where pre-B cells form right around 7 to 8 weeks of gestation. It really isn't until afterbirth that this permanent manufacturing center shifts directly into the bone marrow.
Okay, let's dive into this. Think of the bone marrow as this high-stakes biological sorting facility. It provides the exact microenvironment needed for maturation, it regulates weapon construction, and it basically guarantees each cell has only one specific target. Oh, and it ruthlessly disposes of self-reactive cells. Fun fact for you, millions of B cells are generated here every single day, but only a tiny fraction actually pass these strict exams. The rest, they just drop out.
Let's walk through how this builds across the physical development stages of the cell. We start out with a basic stem cell, and then move through the pro-B and pre-B stages, all the way up to a mature B cell. Now, what really defines each of these distinct physical stages is the crazy activity happening deep inside the cell's DNA. Every stage is strictly defined by the complex rearrangement of immunoglobulin heavy and light chain genes. As those genes successfully rearrange, the cell expresses new surface molecules and takes one giant step closer to graduation.
But here's the thing, graduation is definitely not guaranteed. And that brings us right to Section 2, passing the tolerance test. This is the absolutely critical quality control phase. Why? Well, because a cell that's armed and ready to attack, but aims at the wrong target, poses an immense biological danger to our own bodies.
Before even testing the weapon, the body has to make sure the cell only has one single weapon to test. This introduces a vital concept called allelic exclusion. Essentially, B cells get several chances on their chromosomes to successfully rearrange their genes. If the first chromosome fails, no big deal. It tries the second. But the very moment it successfully builds a functional receptor, it completely shuts down the other options. This guarantees that every single B cell has exactly one specific antigen receptor. Locking in a single specificity is absolutely vital to avoid autoimmune disasters. I mean, you definitely don't want a cell that accidentally targets both a virus and, say, your own heart tissue.
The body really ruthlessly enforces this safety check. If an immature B cell creates a receptor that accidentally targets the host's own healthy tissue, it fails the exam permanently. The cell undergoes clonal deletion via apoptosis, which is just programmed cell death. It's really like a military academy with an astronomically high dropout rate, where failing a single accuracy test means immediate expulsion. Only cells that completely ignore self-antigens are actually allowed to graduate. So here is the crucial question. Our naive B cell has its unique receptor, right? It survived the rigorous academy and it's moved out into the secondary lymphoid organs. But how does it actually know when it's time to drop the resting phase and start attacking? It can't just fire at will.
To answer that, we enter Section 3, activation and T-cell help. For a B cell to unleash its full arsenal, it usually needs to meet its commanding officer to get official authorization for a strike, and that officer is the T helper cell.
Now, what's really interesting about this interaction is the multi-step verification. The B cell presents a piece of an antigen on an MHC2 molecule to the T helper cell. But just recognizing it isn't enough. The CD40 ligand on the T cell has to physically connect to the CD40 receptor on the B cell. That connection right there, that is the critical trigger. Once they're connected, the T cell releases these cytokine spheres, specifically IL-2, IL-4, and IL-5, directly onto the B cell.
Think of it as a complex molecular handshake. The B cell signals via MHC2 and CD40 are essentially telling the T cell, hey, I found something dangerous. In return, the T cell provides the crucial help through CD40L and cytokine delivery.
Those cytokines, IL-2, 4, 5, and others, they are the authorization codes. They completely synergize with the B cell's own signals to promote massive mitosis, differentiation, and isotope switching. Without both sides of this exchange, the whole defense response just stalls out.
Once fully authorized, our B cell undergoes this amazing transformation into an antibody-secreting plasma cell. And that brings us to Section 4, Anatomy of an Antibody. It is time to look at the actual weapons this factory is mass-producing. Immunoglobulins are just brilliantly constructed. They consist of shorter light chains and longer heavy chains, and they're locked together by these incredibly strong covalent disulfide bonds. But the crucial secret to this design? It's the heavy chain that dictates the weapon's class, or its isotope. By swapping out the heavy chain, the antibody becomes IgA, IgD, IgG, IgE, or IgM. Each one is tailored for a completely different theater of war.
Take IgE, for example. It exists in just tiny amounts, literally 0.001% of all immunoglobulins in your blood. Yet its power is profound. It's the primary driver of type 1 hypersensitivity reactions. So if you suffer from seasonal allergies or asthma, IgE is the culprit right there. But its true evolutionary purpose actually isn't to make you sneeze. It's designed to trigger basophils and mast cells to attack massive parasites.
Now that we have manufactured these weapons, it is time to deploy them. Welcome to our final chapter, Section 5, Effector Functions in Action. Let's actually see how these microscopic guided missiles neutralize the threat out in the field.
Here's a great bird's-eye view of the battlefield. Once released, antibodies have multiple distinct effector functions. They neutralize microbes, they opsonize them for phagocytose, they flag infected cells for natural killer cells, and they activate the frankly explosive complement system. Let's zoom in on a few of these crucial tactics.
First up is direct neutralization. On one hand, without antibodies, a microbe easily binds to cell receptors and spreads infection. It basically gets a free pass. But when antibodies are in the mix, they physically coat the microbe. They act like little caps, physically blocking the pathogen from ever binding in the first place.
Now, if a microbe isn't just neutralized, it needs to be completely destroyed. And that brings us to a really crucial piece of medical jargon: opsonization. Opsonization is simply the immune system's way of painting a big, glowing target on the enemy. By coating a microbe with antibodies, particularly IgG, it heavily facilitates and enhances the ability of our phagocytes, our cellular eaters, to just totally consume them.
And this brilliantly illustrates that exact process. The microbe gets coated in IgG antibodies. As it approaches the phagocyte, those antibodies fit perfectly, almost like keys, into the Fc gamma RI receptors on the phagocyte surface. That binding immediately triggers engulfment, dropping the pathogen into an internal chamber where it is rapidly digested and destroyed.
But what happens if the target is just too big to be eaten? Well, that is where antibody-dependent cellular cytotoxicity, or ADCC, comes in. This is exactly where that rare IgE antibody shines. When an enormous parasitic worm invades, IgE antibodies heavily coat its surface. Eosinophils, equipped with high-affinity receptors, bind directly to those antibodies and unleash toxic granules straight onto the worm, destroying a target exponentially larger than themselves. Honestly, it is a stunning display of coordinated biological warfare. Which leaves us with this truly staggering thought. From the rigorous academy of your bone marrow, to those intense life-or-death tolerance exams, to the precise manufacturing of targeted weapons, millions of these complex microscopic battles are being quietly won inside you right this very second. The quiet efficiency of your antibody-mediated immunity is exactly what allows you to survive in a world absolutely filled with invisible threats. But if our internal defenses are this sophisticated, what actually happens when viruses learn to hack the academy itself? Well, class dismissed for today, but keep exploring.